Hello students, welcome to my channel Microbiology Tutorials from Anshul Kumar. Today I am going to discuss a topic about the ARMS-PCR. ARMS-PCR is a type of variant of PCR, and the medical students who have known about the PCR technique would be knowing about the methods of PCR and its importance. So if I discuss about the PCR methods, first of all, it is a type of polymerase chain reaction, and it is used for the amplification of target DNA, and it amplify many DNA cycles through different cycles, many copies of a single DNA, and it identifies the target part. And it is used for in the diagnosis of different type of diseases, including both bacterial and viral infections. The PCR was discovered by Carey Mullis in the year 1983, and for his discovery he was also awarded the Nobel Prize. And the basic process in the PCR is that first the double-stranded DNA is denatured at a high temperature. Then the two strands are annealed with a primer, which acts as a starting point for synthesis of a new strand of DNA complementary to its parent strand. And when again the two strands are converted into four strands, they are again denatured, and further primers are utilized there, and more copies of single DNA are synthesized through this process. Now here, the role of primer becomes very important, because unless and until they will not get a complementary base pair to their parent's DNA, they do not get attached to it. And if they are not attached, the DNA polymerase will not act on the complementary strands and synthesize a new DNA. So basically, so this is the basic concept which is utilized in the PCR step, and when it completes 30 to 35 cycles, so the target DNA can easily be identified through it. Now here comes the fact that when we use PCR, there is a activity of restriction enzymes which breaks down the two strands. But when we are using PCR, we are using an entire DNA genome for the amplification. But the advantage here with the ARMS-PCR is that here even a single allele or single nucleotide have been changed, or we can say a point mutation, either due to deletions, insertions, So these steps, if there is a change in the nucleotide base pairs, it can be also be identified. And here we use 4 primers to detect. So we will discuss in detail about the ARMS-PCR. First, we will see the historical part when it came into this technology was evolved, and it was this was this the scientist who developed this technique, and further it was utilized for detecting some of the important disorders of the body. So the amplification refractory mutation system, actually the term ARMS is an acronym of A stands for amplification refractory. Here refractory means that it resists any type of replication, which is when there is a single nucleotide change in the base pair. Mutation system, PCR. PCR for polymerase chain reaction was originally designed and described by Clive R. Newton and his co-workers in the year 1989, and it was used for known sequence detection of known sequence polymorphism. Clive R. Newton was a lead researcher at ICI Diagnostics in the United Kingdom, who, along with A. Graham and colleagues, developed the amplification refractory mutation system in the year 1989. And this method allows for the detection of specific point mutations and single nucleotide polymorphism in DNA. So regarding the key details about Newton and ARMS, that in 1989 breakthrough, he co-authored the seminal paper on analysis of any point mutations in DNA, the amplification refractory mutation system in nucleic acid research. So what is basically the ARMS mechanism? The ARMS technique uses allele-specific primers that are refractory to amplification, because it means that it resists the amplification. Now here the term comes the allele. So we know that what is the basic difference between an allele and a gene. So allele is a variant of gene. If through example, if I would like to, if I can explain you that gene, suppose a gene describes the nature or the characteristics of any part of the body, just for example hair or color of the skin. But allele can determines whether the color of the skin will be white or black, hair color will be brown, black, or white. So that is the basic difference between an allele and gene. So the ARMS technique uses allele, and if any such mutation occurs in this allele, so it disturbs the entire sequence of the base pair, and it will either lead to the reduction of that particular protein, or the character of the amino acid will change, or it will overproduce the protein, or it may cause alteration in the cell structure due to the mutation. Now it uses allele-specific primers that are refractory to amplification if the target mutation is not present, ensuring high specificity for identifying genetic variations. So here what happens? Here it means that there is a high specific means that false positive results are greatly reduced through the ARMS mechanism. And this technique, often called ARMS PCR or ASP, means allele specific polymorphism, or PCR, is widely used for genotyping, genetic diseases, somatic mutations, and polymorphism. And Newton continued his work on molecular techniques, later co-authoring books on PCR, such as PCR with A. Graham, in bioscientific publishers. Now, the amplification refractory mutation system is an amplification strategy in which a polymerase chain reaction primer is designed in such a way that it is able to discriminate among templates that differ by a single nucleotide residue. Here, I would like to say that the role of the primer is very important in the case of the PCR. It is the primer through which we can detect any type of mutations or change in the primer DNA. Either it is through conventional PCR technique or through ARMS-PCR, and it is able to discriminate among templates that differ by a single nucleotide residue. And the benefit of ARMS-PCR is that it is capable of detecting even a single nucleotide change in the DNA. ARMS has also been termed as allele specific PCR or PCR amplification of specific alleles, PACA. Thus, an ARMS primer can be designed to amplify a specific primer of a multi-allelic system while remaining refractory to amplification to another allele that may differ by as little as single base from the former. The main advantage of ARMS is that the amplification step and the diagnostic steps are combined, that the presence of an amplified product indicates the presence of a particular allele and vice versa. For routine diagnosis, the characteristics of ARMS means that it is a very time efficient method. It is a single step process. It takes less cycle to show the result. However, this combination of the amplification diagnostic step has resulted in a system that may not be robust as some of the other methods in which these two important steps are separated. For example, PCR followed by restriction enzyme analysis. A technique based on the fact that Taq DNA polymerase lacks 3' to 5' exonuclease activity, it cannot extend a primer if there is a mismatch at the very 3' terminal nucleotide. So here the principle beside which is underlying in this process is that usually we know that the activity or synthesis of DNA takes place through the DNA polymerase works in the 5' to 3' activity it takes. But here what happens that since it lacks 3' to 5' exonuclease activity, it cannot extend a primer if there is a mismatch at the very 3' terminal nucleotide. Because it means that if the desired nucleotide sequence, which is in the primer part, and the desired nucleotide sequence, complementary nucleotide is not present on the parent strand, and if we can say it as a mismatch, so there is a mismatch in the base pairing and the synthesis of the nucleotide is terminated at that point. So the term here, two important terms comes, what is matches and mismatches. So when the matches, if the 3' in the such nucleotide sequence are designed, if it matches with the DNA strand complementary DNA strand, the synthesis of the daughter strand will take place. And if it does not matches, or if we can say it, if it mismatches, then extension is refractory and no product is formed. To detect a specific mutation, two separate reactions are typically run for each sample. Normal reaction uses a primer specific to the wild type allele or normal allele, and mutant reaction uses a primer specific to the mutant allele. So here we take two type of primer, one which is primer which is specific to the normal allele, and another primer which is specific to the mutant allele. In addition to this, we take an internal control, a second pair of primers targeting a non-related DNA segments is added to both tubes to ensure the PCR process worked correctly, that it prevents false negative result. Now, what is the primer design strategy? Specific primers designed so the 3' terminal base matches either the wild type or the mutant nucleotide. Common primer, a reverse primer that is the same for both reaction flanking the target site. So two primers are here designed. One is the specific primers, which is designed so the 3' terminal base will match either the wild or the mutant, and a common primer which irrespective of mutation or it is normal will flank to the target site and start synthesizing the daughter strand. Again, what is the deliberate mismatch? Sometimes an additional mismatch is introduced at the second or third nucleotide from the 3' end to increase the specificity and ensure the polymerase fails if the terminal base doesn't match. Here, what happens that here what happens that if sometimes yeah, what happens that if single nucleotide is mismatching, it is possible that it may overcome that error and it will continue to synthesize the alternate strand. But again, if second last or just adjacent to the target nucleotide sequence, if there is a change or that is also not matching with the complementary pair, then the chances of overcoming that error is totally negated. So it is said that an additional nucleotide is added at the 3' end so that if there is a point mutation or mismatch base pair, any possibility of overcoming that error and synthesizing the to the completion is overruled. Now, if we see the result and further after the PCR cycle is over, it is then electrophoresed on a agarose gel and based on the electrophoresis, which we know that the interpretation is done on electrophoretic chamber through or the gel through the appearance of bands. So if the band is present in the normal tube, it is a homozygous normal. If band is present in both normal and mutant tube, then the heterozygous. If band is present in the mutant tube but not on the normal tube, then homozygous mutant are present. Coming to the next slide. So ARMS is a type of polymerase chain reaction that allows genotyping of known single nucleotide polymorphism. For example, normal and mutant alleles. Allele is specific ARMS amplicons are detected by agarose gel electrophoresis. The system is fast, simple, economical since no special lab setup or expensive equipment is required. ARMS can also distinguish heterozygotes from homozygotes for an allele of interest. This experimental system is called ARMS because the normal setup is refracted to PCR on mutant DNA and the mutant setup is refracted to PCR on normal DNA. ARMS concept is based on single PCR with 4 primers under standard cycling conditions, and each allele-specific reaction is prepared for normal and mutant target followed by gel electrophoresis. The alleles are differentiated by sequence-specific primer design where a 3' terminal base is complementary to each allele so that only the target allele is amplified. Allele differentiation is further improved by introduction of a mismatch at the 3' penultimate bases of the primer. Since two mismatch bases at the 3' end of the primer prevents PCR extension and the template contains a non-target allele, in this way, the presence or absence of an allele-specific PCR amplicon is used for detection of single nucleotide polymorphism in a sample. So here you can see that in this way, the agarose gel electrophoresis is done after completion of the PCR technique. So there are two important prerequisites before setting up an ARMS experiment. First, the SNP sequence must be known in order to design allele-specific primers and second is that non-proofreading DNA polymerase, that is without 3', 5' exonuclease activity, must be chosen in order to maintain the 3 mismatch bases in the primers during PCR. Now, what is the difference between the regular PCR and ARMS PCR is that standard PCR amplifies general DNA sequence, while ARMS PCR uses specialized mutation-specific primers at the end to selectively amplify only specific alleles, normal or mutated. ARMS-PCR is specifically designed to detect single nucleotide polymorphism or small insertions or deletions by enabling amplifications only when the primer perfectly matches the target site. Regular PCR amplifies the target DNA sequence for identification or cloning, while it is designed for detecting single base mutations. ARMS-PCR requires specialized primers with an int that matches either the wild or mutant allele. If the int is not perfectly matched, amplification is inhibited. So it relies on the refusal of Taq polymerase to extend a primer with a mismatched end. While regular PCR shows a band if the target exists, ARM-PCR uses multiple primer sets to produce different size bands on a gel, separating the normal and mutant genotypes. ARM-PCR commonly uses nested primers or internal controls to confirm the reaction success, which is crucial for distinguishing between a negative result and a failed amplification. So what is the core principle? So primers are designed so the final base in the 3' end aligns perfectly with the target SNP or mutation. See, basically it is based on the designing of the primers specifically targeting the on the 3' end, targeting the nucleotide at the 3' end. One thing I would like to again add that the mutation or the changes in an allele is basically selected on the either end of the DNA. It should not be central, so that when the steps takes place and primer is added, it will not have to go a longer cycle of, synthesizing the new DNA. It will be it will immediately detect any type of alteration or any type of change in the nucleotide sequence as soon as it attaches with the parent strand. So the mutant strain or change in the nucleotide sequence is not at the central part of the DNA, but basically it is at the terminal part. How the refractory mechanism takes place: if the base of the primer does not match the template, Tag polymerase cannot efficiently initiate DNA synthesis, preventing amplification of that specific template. Sometimes, as I have already told you, then one additional deliberate mismatch is introduced near the end to further reduce non-specific binding and enhance discrimination. Now, two parallel PCR, one reaction uses a wild type, another a mutant primer pair. So what are the key requirements? High specificity, crucial to avoid false positives from low-level primer mismatch extension, especially with high template concentration, as noted in the quantitative amplification refractory mutation system study. Controls, an internal control primer is often required to ensure that PCR failure is not due to degraded DNA. So these are already we have discussed that any mutation which is produced in the allele can be easily detected by ARMS-PCR. In this only, there is one tetra primer ARMS-PCR, which, as the name suggests, uses 4 primers to detect any type of mutations. And these 4 primers are forward outer and reverse outer, which acts as an internal control, creating a large constant band. Next are the 2 inner primers, forward inner and reverse inner. These are allele specific. Means that for forward outer and reverse outer are the common for both normal allele and the normal DNA, as well as the mutant DNA, or the DNA sequence which contains some mutant allele. Whereas the inner primers are allele specific, means that is one that they are one for the mutant and one for the normal, and each is designed with a 3' terminal base corresponding to the target SNP. Mismatch introduction: to increase specificity, an additional mismatch base is often introduced at the penultimate position of the inner primers. Primer should be greater than 30 bases long for better specificity. Amplification refractory mutation system is a simple method for detecting any mutation involving single base changes or small deletions. ARMS is based on the use of sequence-specific PCR primers that allow amplification of test DNA only when the target allele is contained within the sample. Following an ARMS reaction, the presence or absence of a PCR product is diagnostic for the presence or absence of the target allele. The protocol detailed here outline methods that can be used to analyze human genomic DNA for one or more mutations. The basic protocol describes the development and application of an ARMS test for a single mutation. The alternate protocol extends this to multiplex ARMS for the analysis of 2 or more mutations. The support protocol describes a rapid DNA extraction method from blood or mouthwash samples that yields DNA compatible with the type of test described. The amplification refractory mutation system is a simple method for detecting any mutation involving single base change. The amplification refractory mutation system is a simple method for detecting any mutation involving single base change. What are the key aspects of ARM-PCR? So first of all, it is a three primer system. A typical animation shows a common primer, a wild type specific primer, and a mutant specific primer, often in separate reaction tubes. If I can show you through this. So here you can see here this blue one is the allele specific primer, whereas this orange one here, it is the common primer. Okay, so here you can see that if I consider it that the 3' end is complementary to the alternate strand, so what we see that here extension is done by the Taq polymerase. So if it matches here, the nucleotide sequence are matching, they can the extension of Taq polymorphism means that Taq polymerase will start acting. Whereas if in this one, if it is not complementary base pairs, or if there is no matching between the base pair, so since it lacks 3 exonuclease activity, so here there will be no extension. So a standard consists of two complementary reactions and utilizes three primers. One primer is constant, which we have already seen in a tetra, that is the outer, reverse outer and forward outer. And one is the internal, inner and inner, which is the part of the tetra. Similar here also, the same is there. Same is shown here, that two complementary reactions and utilizes three primers. One primer is constant, that is the orange one, that is the common primer, and complementary to the template in both reactions. Another primer differ, they are 3' terminal residues and are specific to either the wild type DNA sequence or the mutated sequence at a given base. Only one of these primers is used per tube. PCR products are separated by electrophoresis when agarose gel containing ethidium bromide or fiber green. The sample is homogeneous mutant or homozygous wild type. Amplification will only occur in only one of the tubes. If the sample is heterogeneous, amplification will be seen in both tubes. So here basically we use 50 to 100 nanogram genomic DNA master mix is 1x PCR master mix, which contains Taq polymerase, deoxynucleotide triphosphate, magnesium chloride, and buffer. And primers are usually used in an optimized ratio. Generally, inner primers are added at higher concentration than outer primers. For example, 1:2 or 1:3 ratio. A common ratio is 0.1 micron inner and 0.05 micron outer. PCR amplification, basically initial denaturation at 95 degrees centigrade for 2 to 5 minutes, cycling 22 to 35 cycles, then denaturation, and again 95 degrees centigrade for 30 seconds, annealing optimized at 54 to 69 degrees centigrade for 30 to 60 seconds, and then extension at 72 degrees centigrade for 1 minute. And the process is simple and effective, and hazardous radio level probes aren't involved here. We can divide the whole process into 4 separate steps. This primer designing, which is one of the most important steps, then amplification, agarose gel electrophoresis, and then result interpretation on the basis of band formed on the electrophoretic chamber or electrophoretic gel. So basically the gel which is used are the 2% agarose gel stained with ethidium bromide or cyber safe, visualized under UV light. Results homozygous. If there is an homozygous mutation, then large control band plus mutant band. If homozygous wild type, Homozygous means that both the alleles which has been obtained from the parents are the same, and heterozygous means that the allele that is different. I can say that if two capital B are on the present, that is homozygous, and one capital B and one small b, then it is called as the heterozygous. So homozygous wild type, large control band plus wild type band. Heterozygous, all three bands control, mutant and wild type. What is troubleshooting and optimization? Annealing temperature, perform gradient PCR to find the optimal temperature for specific amplification. Primer concentration, adjust the inner outer primer ratio to prevent a single band formed from dominating. And false positive, reduce the number of PCR cycles and ensure strict controls. So these are some of the important steps, and it is one of the important technique to diagnose some of the hematological disorders, especially thalassemia conditions, as well as it is also used in detecting some of the important cancerous conditions in which there is a single point mutations. So all such and many more uses are there of this type of PCR. So it have revolutionized the medical diagnostic techniques, and it is one of the important molecular technique which can used in a very cost effective and rapid manner in comparison to conventional PCR. So with this, I conclude my lecture. I hope you would have certainly benefited from it, and please do like and subscribe my channel so that I can continue bringing new topics and innovative topics discussing about the modern medical sciences. With this, I conclude today's lecture. Thank you.